Well, hello. This is lecture 8 for our microbiology course. This is where we are going to focus on host microbe interactions as well as the science of epidemiology. This is going to cover chapters 16 and 19. Now, host microbe interactions are going to be pretty straightforward in the sense that this is kind of following after lecture 7, which was on the immune system. Epidemiology is going to take this and how many of these diseases are spread, etc. Now, we're going to start off with chapter 16. And this is really important. I really encourage students to take the time to read the key terms as well as the glimpse of history. One of the things you have to keep in mind when we're talking about all of this is that normally we have normal flora, organisms that routinely reside on the body's surfaces, okay? Keep in mind that some of the organisms provide benefits. Others are harmless, as their capacity to evoke disease has been kept in check. When we talk about a pathogen, this is a disease-causing organism or agent. When we talk about someone immunocompromised, also known as immunologically compromised, these are individuals whose immune system defense, both or innate and adaptive, so basically it could be either or both, and usually it's both, is weakened or defective. Thus, the individual is vulnerable to invasion, which will cause disease by other organisms, including those from the normal flora. Keep in mind that it's almost a balance of power, if we want to say, between normal flora and our immune system. And when the immune system weakens, some of the normal flora may then cause actual disease. Also, you want to keep in mind that weakened immunity can occur through malnutrition, cancer, AIDS, genetic defects, alcohol or drug abuse, surgery, or immunosuppressive therapy, such as drugs, radiation, etc. Now, when we go over the anatomical barriers, we have discussed this before. The body is a complex ecosystem for microbes. When we talk about the term symbiosis, this is the process of biological organisms living together. There are different forms, though, of symbiosis. First, mutualism, the association where both partners benefit. Okay. Commensalism is an association where one organism benefits, the other is neither harmed nor helped. Finally, parasitism. This is the association where one organism is harmed, the other one is helped. So in parasitism, one organism is benefiting, the other one is being harmed in some way. Now, let's go through what we would consider normal microbiota, and you can see here from this diagram, you can find them in nose, mouth, throat, large intestine, urethra, vagina. Normal microbiota, and they used to call it the microbiome or normal flora. And this is the population of microorganisms found growing on the body of healthy individuals. There are two types: resident microbiota, and these are microbes that inhabit body site for extended periods. Transient microbiota are microbes that only reside on a temporary basis. Now, the metagenomics, this is the analysis of DNA extracted directly from a given environment, allows us to measure the types and activity of the resident microbiota, the transient microbiota, etc. And some of these insights have given us a lot of much better understanding of how these microbes can cause disease and how we are able to defend against other disease pathogens. But first off, let's start at the beginning. During birth, babies acquire lactobacilli through the passage in the birth canal. They reside on the skin and in the digestive tract. Now, here's an interesting question. Well, gee, if they didn't go through the birth canal, what about if they went through C-section? It's interesting that they have found that cesarean section babies have different microbiota. Some doctors will actually, after the birth, try to inoculate the children with some of the I shouldn't say pathogens, but the microbiota from the vagina or from a culture, just to help them. Now, adult microbiota populations include firmicutes, which these include Clostridium and Bacillus species, and Bacteroides, which of course, these include the Bacteroides. Now, the balance of the species in the digestive system has been found to affect metabolism and body weight. As a matter of fact, it's been found that obese versus thin individuals have different compositions of microbiota in their small intestine and large intestine. What are some of the beneficial roles of normal microbiota? Well, first off, protection against infection. When normal flora are killed off by antibiotics or their growth suppressed, then other organisms can occupy the site. The mechanism to exclude pathogens by normal flora include the following. The flora will consume nutrients that would normally be available for pathogens. They cover binding sites for pathogen attachment. They produce toxic compounds. Lactobacillus species will suppress the growth of Candida albicans in the vagina because lactobacillus will release acidic lactic acid. That drops the pH and makes it hostile for Candida albicans to proliferate. They also will prime the adaptive immune system. And that's where you're getting into the Peyer's patches and other malt tissues. Remember, mucosa associated tissues that you'd find in the small intestine. Now, another thing that the microbiota do is they promote oral tolerance. They teach the immune system to tolerate various antigens. This is where we get into what's called the hygiene hypothesis, which proposes that insufficient exposure to microbes can lead to allergies and autoimmune diseases. This has been found. You know, how come the kids that grow up in the city versus the kids that grow up in the country on farms have differing amounts of allergies and autoimmune diseases? Basically because they've already been exposed to them. This also kind of explained why, well before the polio vaccine was out, they noticed that children in dirty environment never got adult polio. Well, it's because they were exposed to polio when they were very, very young, fought it off, and thrived. And then you have adults living in the city in ultra-clean environments, etc., that get exposed to the poliovirus later, and it proliferates in them. That's a hypothesis and an attempt to explain some examples, but doesn't mean it's the end-all and be-all in conclusion. Also, the microbiota will aid in digestion. For example, some microbes break down dietary fiber. Others will produce substances important in human health. Bacteria in the digestive tract produce vitamin K. They will also produce select B vitamins, such as niacin and biotin. And this is absorbed and used by the host. So you have sort of a mutualistic relationship there. It is conceivable that as the years go on, we'll probably see more revelations of this. You probably already see this in the supermarkets and in the drugstores under the labels of probiotics. Some of them can claim a lot, but others have the research to back up their studies. Now, the dynamic nature. From birth, humans are exposed and colonized by microbes. At the gut, the skin, the vagina, the microbe ecosystems can be disrupted by hormonal shifts, the nutrition of the human host, use of antibiotics, etc. When we talk about the principles of disease, we need to get familiarized with a series of terms. For example, infection. When a microbe has a parasitic relationship with the host. When we talk about colonization, this is when a microbe has become established on the host and it is multiplying on the body surface. You need to note, though, that infection does not always lead to noticeable adverse effects. Symptoms may be subclinical or unapparent. Symptoms, these are effects of the disease experienced by the patient. Signs, these are the effects of the disease that can be observed by examining the patient. So there's a difference between symptoms and signs. Okay? Also, primary infection. That's the initial infection. Secondary infection is an additional infection that occurs as a result of the primary infection. Now, that may be sounding very confusing, but as we go through the up and coming weeks and give you a variety of examples, you should get a much clearer picture of what we're talking about here. Pathogenicity. A primary pathogen is a microorganism or virus that can cause disease in an otherwise healthy individual. Okay? It's still a pathogen. An opportunistic pathogen, otherwise known as an opportunist, is a microbe that causes disease only when the body's innate or adaptive immune defenses are compromised. Organisms can be present in the environment or part of the normal flora. So as we talked about earlier, if you start having the immune system starting to falter or weaken, then opportunists can actually take over and become actually a pathogen. The concept of virulence refers to the degree of pathogenicity of an organism. This reflects both the proportion of infected people who have obvious symptoms and the relative severity of those symptoms. A highly virulent organism has more disease-promoting attributes than less virulent strains. Think about it this way. There's some species, in short, what I'm trying to say, that have a great deal of capacity to cause disease, damage to tissue, things like that, and some others do not. The example that's provided in the notes is that Streptococcus pyogenes, some strains will cause strep throat, while others are much more virulent and can cause necrotizing fasciitis. Basically, you're all familiar with that flesh-eating disease. It has greater amount of toxins and enzymes, etc., that can cause the destruction of the layers of the fascia, which is below the skin, as well as, of course, the skin. What are some characteristics when we talk about infectious disease? Well, they're communicable, or a contagious disease, when we talk about that. It's an infectious disease that can be spread from one host to the other. This is easy. Flu, colds, measles, etc. But the term infectious dose is something you need to know about, and that is the number of microbes necessary to establish an infection. Some diseases require many microbe cells, as many are destroyed via stomach acid, antibodies, and other innate and adaptive defenses prior to the infection being established. Others require only a few cells, 10 to 100 cells, for example, for Shigella. We come up with the concept ID50, which means infectious dose 50. This is the dose of cells where 50% of the population develop the disease. When I was doing my studies for bioterrorism and biowarfare, there were some organisms that if you just gave 10 cells to the individual, they would get the disease. Some also talked about, for example, well, you could have higher numbers of this and higher numbers of that, but really, Bacillus anthracis, if you get one of them into the lung, you pretty much will get anthrax. Now, some of this is because they have the capabilities to defend themselves against the host defenses, which we'll get into in the next, in the later parts of this chapter, I should say. Now, we've been talking about that in virulence factors. Oh, I missed that. I need to go over that. These are traits on an organism, or traits of a microorganism that specifically allow it to cause disease. If you remember before, we talked about plasmids. Genes for such traits can be transferred to other bacteria via horizontal gene transfer. They may exchange from one bacteria to the other plasmids that contain particular genes that would enhance disease capabilities or disease-causing capabilities to organisms. Bear with me, I want to make sure that you get everything here. Now, when we talk also about principles of infectious disease, we've got to talk about the course of infectious disease. Now, this is where you see the chart up here. Incubation period, this is the period between the introduction of the organism until the onset of the illness. The illness is referred to as the period where the patient exhibits signs and symptoms of the disease. When we talk about convalescence, the stage of recuperation and recovery from the disease. We also have to deal with carriers. These are individuals that harbor and may even spread disease organisms, even though they show no signs or symptoms of the disease. This may actually last for months or years. Oh, and just one other important thing. Incubation period. For some people, they think, oh, I'm exposed and tomorrow morning I'll get it. Sometimes it takes a while for the particular pathogen to build up. And meanwhile, while they're building up in numbers in the patient, they may now at the same time be contagious and be able to spread the disease, even though they haven't shown disease symptoms, even though they don't feel sick yet. I want to make that clear. It depends on the pathogen itself. As for the duration of symptoms, infections and disease are often described according to the timing and duration of the symptoms. When we talk about an acute, these are symptoms that have a rapid onset but last only a short period of time. For example, cold, 7 days. Chronic, these are infections that develop more slowly, tend to last much, much longer, perhaps months or years. An example of that would be tuberculosis. And then we have latent. Now, latent infections, they never really are completely eliminated. The microbes tend to remain in the tissues, sometimes for years without causing any symptom. Sometimes an event or weakness in the immune system will allow for the reoccurrence of the illness. Examples of that are cold sores. You may have shingles, even though when you were much younger you had gotten the chickenpox, genital herpes, typhus. When we talk about the distribution of a pathogen, this is an infection that can be described basically depending on the location of the body. So, for example, if it's localized, the microbe is limited to a small area, Staphylococcus aureus, in the boil of on the skin. Systemic or generalized, this is where the infectious agent has spread or disseminated throughout the body. For example, measles. Now, whenever you see the suffix emia, emia, this means it's in the blood. Case examples: bacteremia, this is bacteria that are now circulating in the bloodstream. Toxemia, toxins are circulating in the blood. Viremia, this would be viruses that are circulating in the blood. And of course, septicemia. This is acute or life-threatening agents or their products circulating in the bloodstream. Okay? Now, we're going to get into in a few minutes, and you can see this other chart which I included to give you a different perspective about the difference between acute, chronic, and latent. Let's now get into causation postulates. Now, you have to understand the importance of Koch's postulate. Before Robert Koch had developed this postulate, before he was able to really clearly show certain principles that were sort of the gold standard to define if an organism is a pathogen or a disease-causing agent, people would come up with all sorts of crazy explanations. They thought that it was bad air or impure odors that cause disease. Well, if you think I'm kidding there, the definition of malaria, everyone understands. It is a protozoal parasite transmitted by a mosquito. Go back some centuries, malaria meant bad, air. In other words, a disease that was caused by bad air, maybe because you were close to swamps and got swamp air. This is crazy as we would think now, but that's where some of this stuff came from. So what happened is Koch had developed these methods to really clearly establish whether a microbe was in fact pathogenic, in other words, causing the disease. This occurred in the 19th century. He established a set of rules for this type of identification. It's used for identification of Bacillus anthracis as the agent of anthrax. You can see this on figure 16.3. His postulates are as follows. 1. The microorganism must be present in every case of the disease. 2. The organism must be grown in pure culture from diseased hosts. 3. The same disease must be produced when a pure culture of the organism is introduced into susceptible hosts. 4. The organism must be recovered from those experimentally infected hosts. Now, of course, today we have certain ethical challenges because you have the problem of when pathogens only infect humans. In other words, you don't go down and take a bunch of people and say, I'm going to verify that HIV is causing AIDS by injecting everybody here with HIV. And that would not go well. No. So you have to do other techniques that would try to demonstrate this. Now, I also bring up to you what's called the molecular postulate. This came in more the 20th century. This is a group of criteria that was set up using molecular techniques. Okay, so we're talking about gene identification, biochemistry, etc., to help identify strains that have virulence factors. The main point is that the genes of different strains of an organism are more important than the species in determining the pathogenic potential of the organism. Give you an example again. E. coli. There's some E. coli that's totally harmless, and then there's others that cause serious disease and can kill individuals. What's the difference? They have certain virulence factor genes in them. So let's go over the postulates. Number one, the virulence factor gene, or its product, should be found in the pathogenic strains, but not in non-pathogenic strains of the suspected pathogen. Number two, introduction of a cloned virulence gene should change a non-pathogenic to a pathogenic state, whereas disrupting the function of the virulence gene should reduce the virulence of the organism. Okay, so think about this. You put in a plasmid containing that virulence factor gene, and the bacteria, bang, become the nasty disease. Or you can neutralize the effect of that plasmid, and you revert it back to non-pathogenic. Number three is the genes of the virulence must be expressed during the disease process, and that's a key point. You have to identify, is this gene actually being expressed? And then finally, antibodies or immune cells specific for the virulence gene products should be protected. To give you a heads up, I really think you need to memorize cautious postulates. I'm not going to hold you necessarily to all the molecular postulates, but it's going to help you to understand some of this by trying to take and review this for cautious postulates. Now, the mechanisms of pathogenesis, excuse me. When we talk about mechanisms of pathogenicity, the methods are basically by which the pathogen evades the host's defenses or causes damage. Now, there are a lot of them. And when I talk about virulence determinants, this is the arsenal of mechanisms that an individual pathogen possesses. And we really need to get into this because you have to keep in mind, as previously discussed, the DNA encoding the virulence factors can be sometimes transferred to other bacteria, to completely different species. Okay? Case in point, antibiotic resistance. Balanced pathogenicity. This is the relationship between the host organism and the pathogen. The host-parasite relationship develops over time, by which the pathogen, or they also call it the parasite, does minimal damage to the host. This occurs as the pathogen develops strains that do little harm to the host, but allow the pathogen to maintain presence inside or outside of the host. It seems kind of weird, but you have to think about this. If you were a balanced pathogenic agent, what happens is that you create the conditions to thrive, you keep your species living inside of the host. You do as minimal amount of damage as possible, but are able to maintain living there. Now, we need to start talking about mechanisms for this pathogenesis. The mechanisms by which a microbe causes disease are any of the following. Now, as I go through some of them may also come to your mind aside of what I've listed. Hang in there. We're going to have a lot of fun as we go through some of this in the latter third of the course. For example, the production of toxins that are ingested. An example of foodborne intoxication, food poisoning, these include Clostridium botulinum, which is botulism, Staphylococcus aureus, which causes staph food poisoning, Claviceps purpurea, this is fungi, which creates egg rock poisoning. Also, we have the colonization of the surface of the host, followed by toxin production. Corynebacterium diphtheriae, which produces the diphtheria toxin. Invasion of the host tissues. An example of that would be, Mycobacterium tuberculosis or Yersinia pestis. Yes, that's basically the Black Death, otherwise known as plague. Invasion of host tissues, followed by toxin production. There you would get Shigella dysenteriae. Now, when we talk about infection methods, we talk about several different mechanisms. It sounds crazy when you think about it because bacteria have developed these strategies, but this is how we found some of these things that actually do work. I turn your attention, for example, to figure 16.4, for example. And what you see, let me go back here for a second, was the adhesins that, with the pili that allow the bacteria cell to attach to the host cell. So this is adherence. The microbes will use the means to attach to host cells. To bind to the host cells, the bacteria will use these adhesins, which are proteins on the cell. They allow the bacteria to attach to the host cell, and these adhesins are located on the pili. So the pili are going to be used for attachment, and when they do this, they're very often also called fimbriae. Some bacteria have special receptors that attach to specific cell receptors. When we go to the next area, we talk about colonization. Now, during the growth and development of bacteria on a host, bacteria must compete for nutrients and protect against IgAs, which would be the immunoglobulins type A, the antibodies found in the mucus and in the GI tract. Now, IgA defenses can be assisted as some bacteria secrete IgA proteases, which destroy the IgA. What did I just say? Basically, how are you able to defend against an IgA attack? You have an enzyme that breaks them down. And so you were able to thrive there. Also, bacteria cells perform antigenic variation and alter the pili produced to evade immune defenses. What am I saying there? In short, think of it this way. Some bacteria act almost like chameleons. They will shift their antigens on their surface. And by the way, this doesn't just happen with bacteria. We can also see this happening with certain helminthic parasites too, and we'll talk about that later. Because they change the antigens on the surface, they can now evade having antibodies attached to them, etc. Now, some pathogens actually will produce iron-binding molecules. These are called siderophores. They sequester iron from the host proteins. And finally, type 3 secretion system. This is also known as an ejectosome. This is where you have gram-negative bacteria that use proteins delivered into host cells to alter the shape and function of the host cell to favor uptake by the bacteria. What you see there is that it looks like almost an injector pump. You see the effector, the bacterial cytoplasm, and the periplasm. You see this site, and you on the right, you actually have an electron microscopy image of this type of protein, and it will inject into the host cell what is necessary. Now, when we talk about invasion, this is breaching the anatomical barriers, and this is going to get really interesting. So what happens? Now, different pathogens have differing strategies to breach the anatomical barriers. This is part of the innate or passive immune defense system. Remember, we're talking about breaching, let's say mucous membranes or skin. And keep in mind something too. It's not just your skin, it's not just your mouth, nor your nose that you can get infections. Doctors found out later on, and that's why they wear goggles when they do surgery, because the eye has become a portal of entry. A speck of blood coming from the patient while they're treating it can actually carry sufficient, viruses or bacteria, and the doctors will get an infection. Now, we also have to keep in mind that one of the other ways to breach the anatomical barrier is penetration of the skin. Some pathogens can only get into the skin by either injury or abrasions. Others will get injected via an arthropod bite or a puncture. We'll get into wound injuries down the road, and one of the interesting ones is when you get a bite, either cat, dog, or human. That gets interesting. We'll talk about that later on. Now, another means of invasion is penetration of mucous membranes. Two methods. You ready? Directed uptake by an epithelial cell. The pathogen induces endocytosis of the microbe into the host cell. Sometimes This is the ruffling, which is a directed disruption of the cell cytoplasmic membrane due to the cytoskeleton breakdown occurring. Now, what you see here in this diagram is a little bit of this. This is a situation where literally you have injected the secretion system, you have a bacterium, and what it does is the microvilli collapse. Remember, microvilli are held up in part by actin microfilaments. Now you've got this flattened area that allows for a better contact and hopefully greater injection into the cell. Here you have a ruffle and a bacteria cell. Salmonella enterica, and this is the serotype Typhimurium, is inducing ruffles. In other words, what they're doing is they're flattening out and making a big, almost like landing pad. And this is occurring on an M cell, which is a specialized intestinal epithelial cell that will allow for the uptake of the bacteria cells. Alrighty? Now, you see this as antigen sampling process, which provides for another mechanism of infection. But let me just finish this up here. When we talk about directed uptake, the Shigella invasion of the M cell of the intestine, which I just showed you as an example. Another one is the exploitation of the antigen processing system. Here, you're going to use the M cell again, transcytosis process. The bacteria enter macrophages. They will evade digestion. They will destroy the macrophage. They will escape, and they will enter other mucous membrane cells. You can see this here through a 1-2-3 attack. And by the way, what you see when you get into part 3, Shigella has this unique capability to take the host's actin proteins, make them into a big long tail. They polymerize them, and this gives them the propulsion for the bacteria cell to literally make it sort of like a downward sloping train track and helps push them right in to neighboring eukaryotic cells in the intestinal lining. That's why it's so nasty. It'll start destroying cells, and it spreads out, invading cells that are nearby. Now, how do we avoid all of this nastiness? Oh, and by the way, here's a great photo of that. That's okay. You can say I don't have a life. I understand that. That's fine. You see the green, that's the actin tails. And in this case, it's Listeria monocytogenes that are doing this rapid intercellular propulsion setup by the bacteria. The bacteria are kind of the yellow or reddish orangish, okay? When we talk about avoiding host defenses, okay, just because you're in the cell doesn't mean you're not going to face all of that. And you have to find various strategies to protect yourself to continue the infection. To give you a background, just as a simple thought, ask yourself this question. What do I need to do to survive? I know you and I would sit there and say, well, I got to make a paycheck, I got to eat, I got to do this, I got to do that, yeah. Most other forms of life, even those that are pathogenic, are having maybe not the conscious thought, but they are having the mechanisms that basically ask the same question. What do I need to do to survive? So when we talk about avoiding host defenses, how does a cell evade the host immune mechanisms to set itself up for an infection? The more I can do an infection, the more I can reproduce of my species. The more I can reproduce of my species, the sustainment of the species as a whole. But so what if the host suffers? Different pathogens have developed various strategies to avoid host cell immune defense mechanisms. One of them is plain out just hiding. You can hide inside the host cell. This avoids antibodies, phagocytes, complement defenses. Movement within the host cell can occur via actin polymerization to propel the pathogen through the host cells. Well, golly gee, we saw that a minute ago back here. Shigella, Listeria monocytogenes, they do the same thing. The next one is defense against complement proteins. Excuse me. Now, complement proteins cause opsonization, inflammation, and cell membrane puncture. Bacteria have developed cell substances like silagic acid that bind to the complementary regulatory proteins and they block the complement protein cascade. So they stop it from forming and stop it from forming, for example, that pit or hole that they're going to puncture the cell wall. Okay? Gram-negative bacteria that circumvent this process are called serum resistant. Serum resistant. You need to know that term. You can also just plain old avoid phagocytosis. How do you do that? Well, first way is avoid attachment by the phagocyte. And how you do that is with S-pyrogens, you make an enzyme that degrades the complement C5a, so it would be called C5a peptidase, and it blocks chemoattractant chemicals. S-pyrogens also makes streptolysin O, which damages the phagocyte membrane. So even if you get trapped inside, basically you damage the membrane and create instability to try to free yourself and stay inside the cell but not digest it. Some cells use capsules, such as you see here. And some have an M protein, which blocks complement cascade, or Fc receptors, which attach to the Fc region of the antibodies, and they block phagocyte attachment, which you see here. Okay, 1, 2, and 3. Now, how else do I survive within the phagocyte? Even after being taken inside the phagocyte, some pathogens have defenses to prevent cellular digestion. So you escape from the phagosome, the vesicle inside the phagocyte. An example of that would be Listeria monocytogenes. Yes, I do trip over my tongue once in a while, forgive me. Preventing the phagosome-lysosome fusion. An example of that is Salmonella. Surviving within the phagolysosome. Well, Coxiella burenti, which if you remember, that is a very specialized obligate intracellular organism. This is the agent that causes Q fever, does this very well. Another thing that you can do is, besides disrupting opsin, is that you can basically avoid antibodies altogether. Wait a minute. You can't avoid them, but you can avoid attachment of the antibodies. Different microbes use the following strategies. They have IgA protease, which, remember, that's an enzyme that will break down IgA immunoglobulins. Antigenic variation. They basically switch the genetic profile of the surface antigens. As I said, the example of this would be, suddenly being a chameleon. First you're painted black, then suddenly the black paint changes to white. Or you've got flags over you that are of an A protein, and you suddenly switch them and they become B protein. And by changing the antigens, it makes it that much harder for any antibodies to bind onto, or for any actions to be taken by the rest of the immune system. Mimicking host molecules. Mimicking host surface molecules to avoid binding by antibodies. An example of that is S-pyrogens. They happen to have a capsule composed of hyaluronic acid, which is a polysaccharide found in tissues. Okay. Now we start coming up into this incredibly large table. And this is where we talk about damage to the host. This is a list that is vast. Now, before you start hyperventilating or saying, I'm going to lose sleep or I'm going to lose everything, you don't need to. I'm not asking you to memorize that entire table. Maybe if you were an immunologist or you were an infectious disease specialist, that sounds cool. I do encourage you to read over it somewhat. You're going to eventually get reintroduced time and time again to the various pathogens as we go into the latter third of the course. But you really have to see some of these and the different groupings and classes that they are and how they can damage the system. Basically, to cause disease, many pathogens must evoke some type of damage to the host. Some of the damage facilitates for the dispersal of the pathogen to other hosts. For example, when you've got Vibrio cholerae, which is basically the pathogen that causes cholera, you tend to produce 20 liters of microbe-laden watery diarrhea a day. Now, that's spreading cholera everywhere, and that's why it's so incredible to contain once the disease has hit, because people are basically letting out a lot of microbe-laden, what they call rice water, out every day. Bordella pertussis, when you have the violent coughing, this creates a large dispersal of airborne pathogens. Now, the damage due to the infection can be either the direct effect of the pathogen, the effect of the pathogen toxins, or pathogen-initiated over-response of the host's immune system. Let's take a look at Table 16.1. Some terms you need to be aware of. When I talk about an exotoxin, these are proteins secreted by pathogens that have specific damaging effects. They may act either locally or systemically. When I talk about passive immunity to pathogen toxins, that can be provided by an antitoxin, basically antibodies specific to the toxin. Active immunity to the toxin can occur via a toxoid, which is a toxin-containing vaccine. Toxoid may contain a denatured toxin protein or a fragment of that toxin. Neurotoxin, a toxin that damages the nervous system. Enterotoxin, a toxin that damages or causes damage or disruption to the gastrointestinal tract. For example, diarrhea or vomiting, things like that. Cytotoxin causes lysis of the cell or interferes with essential cellular activities. For example, Corynebacterium diphtheriae, which causes the diphtheria disease, releases an exotoxin in the throat. The local cells are destroyed, resulting in the formation of that pseudomembrane. And by the way, that pseudomembrane is nasty. We'll see a photo of that later on in subsequent chapters, but it can also block the airway. The toxins can be transported to other organs such as nerves, brain, heart, kidneys, and damage those organs as well. Now, consisting of the three major classes for toxins we will be dealing with here are the AB toxins, the membrane-damaging toxins, the superantigens. There are a few of the toxic proteins which we'll mention toward the end. AB consists of two subunits. Okay? Here's another diagram here. Usually, when you have an AB toxin, part A is the toxin, part B is a binding protein that binds to specific receptors on the cells. The membrane-damaging toxins disrupt plasma membranes of cells and cause the cells to lyse. Okay? We haven't gotten there yet, but we'll get there. Hang in there. S. pyrogens toxin, streptolysin O, causes beta hemolysis of red blood cells. Oh, wait a minute now. So that makes sense why we have a blood agar culture. If we have the red blood cells present, we would see that if you have the presence of this toxin streptolysin O, you'll end up with the red blood cells bursting and having a unique formation on the presence of that agar. Right. Also, you have phospholipases, which act to remove the phosphate head of the cell membrane. Remember, phospholipids on the cell membrane, you've got that phosphate part, which is sort of facing the hydrophilic environment, and then you have the nonpolar fatty acid tails that face the nonpolar end, the inside of the membrane. So when you start lopping off all of those phosphate heads, the membrane becomes unstable. Now, by the way, the phospholipases is an alpha toxin of Clostridium perfringens. This is what causes gas gangrene. When you start talking about superantigens, you see down there, these incite toxic release of cytokines by activating excessive numbers of effector T cells. Superantigens can cause the situations where individuals will have what they call a superinfection. Individuals can have what they also call cytokine storms. They basically, in some cases, can have their lungs fill up with fluid as a result of the infection in the lungs. Now, endotoxins are a little bit different. Oh, sorry about that. And endotoxins, example, lipopolysaccharide that is present in the cell wall of gram-negative cell walls. This consists of lipid A. If localized, can help the innate immune system clear the infection. But if systemic, such as with septicemia, this can trigger inflammation throughout the body, extensive leakage of blood fluid into tissues, and activation of coagulation. Okay? So what you're talking about are some serious conditions here. Fever, a serious drop in blood pressure, and also what is referred to as disseminated intravascular coagulation, DIC. Basically, you're having clots form all over in small and large areas throughout the body and various organs. Usually, this has been referred to as septic shock. Assays to detect endotoxins use Limulus polyphremnis. You've probably never seen that before, but the next time you see the fishing fleet come in on Cape Cod, take a look. They might be doing horseshoe crabs, because that's what Limulus is. They take the blood from these horseshoe crabs. Pharmaceutical firms that make IV liquids, etc., and it is the blood that will coagulate. And this test is accurate to 10 to 20 picograms of endotoxin per milliliter. Okay? We're talking about somewhere in the trillionth or quadrillionth, exceedingly small amount. Other toxic proteins, exfoliatin. If you talk about exfoliatin, this is produced by some strains of Staphylococcus aureus. It destroys material that binds together the layers of skin, literally causes the outer layers to peel off. This is where you're going to get the scalded skin syndrome. Okay? The toxin spreads systemically, and you'll see that later on as we talk about some of this infections in the skin. What about, and by the way, I encourage you to take the time to go back over and review comparison of exotoxins, endotoxins, okay? It will help you to understand them and generally make some differentiation that'll help you later on. The damaging effects of the immune system are a little bit more interesting from this perspective. During the infection and during the process to remove the pathogens, the host tissue may be damaged as well. Much of the subsequent damage to the host tissue may be due to the after effects of the pathogens. Inflammation. Some inflammation may occur after the phagocytes release their enzymes and toxins used in the innate immune defense. Antigen-antibody complexes. These are some complexes that form from pathogen antigens and antibodies. These complexes may cause inflammation wherever these complexes settle, kidneys, joints, etc. This can cause acute glomerulonephritis. Glomerulonephritis is a nasty situation, and this can follow a Streptococcus pyogenes infection. And as the complexes settle into the kidney glomeruli and damage these structures. I haven't finished yet, but I'm going to get up to there. What I wanted to do was just bring this up. We will be revisiting it later on when we get into infections in the general urinary system. Also, we've got to deal with cross-reactive antibodies. Some antibodies are designed to attack Streptococcus pyogenes, and this is the strep throat infection. But they're similar to normal tissue, to certain MHC types, and this leads to autoimmune attacks on tissues with the MHC marker. This is why sometimes they have to be watchful for strep throat infections that can lead to damage to the heart and other tissues. Now, what about viral pathogenesis? Now, I'm going to go over through a few of these because I really want you to have some appreciation for this. Okay? Now, how do viruses attack and invade host cells? Well, you got to keep in mind the viruses must bind to the host cell. Then they enter. There's no active energy used by virions outside of the cell. So viruses must evade the immune system, and yet they must use the host cell receptors as attachment sites to come in contact with the host cell, as well as attempt insertion into the cells. Once the viruses have reproduced inside the host cells, Then, upon release from that cell, they may enter other host tissue cells, switch from the GI tract to travel in the blood, to the nervous system, to attack motor neurons, as is in the case of polio. How do you see them avoiding immune response? Well, viruses may have mutations which allow their surface proteins to change to avoid immune system. Also, some viruses interrupt an infected cell's production of interferon, the cytokine, and a signal for an infected cell. This stops the cell from being destroyed by the innate immune response. Also, regulation of the host cell death. Some viruses will actually block apoptosis. Yeah, they will actually block the programmed cell death, so that the viral replication will continue, but the cell does not self-destruct. Viruses may also force the development of newer versions of MHC cell receptors to block the natural killer and T cytotoxic cells from destroying the virally infected cell. Antibody defenses of viruses? Well, keep in mind something right off the bat. If you're inside the cell, the virus is safe. You're not going to have the virus dealing with antibody binding. Some viruses cause cells to fuse together to form a super multinucleated cell called a syncytium. Finally, viruses may have error-prone RNA polymerases. These are particularly seen with RNA viruses that create new mutations and thus new strains of the virus. As the immune system cannot adapt that rapidly to the new strains, and antibodies cannot bind to the new viral antigens, basically the viruses are free to exploit the immune system weaknesses to adapt to block the infection. And another way to say it is this, and this is very important. We talked a little bit before about antigenic shift and antigenic drift. When you have a drift because of mutations, part of that may be because of the sloppy RNA replicase, a polymerase that's occurring, because it will tend to create new viral versions with these slight errors that can create a new strain that can evade the human, let's say human, immune system. Okay? Let's talk about eukaryotic pathogenesis. Now, although basic steps of pathogenesis of eukaryotic cells are very similar to the bacteria, colonization, evasion of host defenses, invasion, and damage to the host, some of the mechanisms are really not that well understood. For example, with fungi, many fungi are saprophytes. They consume nutrients from dead or dying organic tissue, and some of the fungi are parasitic. When you deal with dermatophytes, these cause superficial infections to hair, skin, nails, but they don't invade deeper tissues. Keep in mind something. What's hair, skin, nails? If you remember your A&P, it's all basically dead cells when you get outside of the skin layer. These fungi are basically using keratinase enzymes. They degrade keratin, and they result in what we call ringworm and athlete's foot. Some normal flora, which infects immunocompromised patients. You have Candida albicans, and this leads to infections in the throat, mouth, can also lead to vaginitis. This is a big problem with patients that are AIDS patients or burn patients, okay? Cryptococcus species can make a large capsule that interferes with phagocytosis by immune cells. Now, the most serious fungal infections come from the dimorphic fungi. Remember dimorphic? They can appear as single cells in the yeast format or as filamentous as molds. This is where you get a situation where some of these dimorphic fungi, they occur as molds in the environment but act as yeast in the host organism. Usually inhaled as spores, these fungi are ingested by lung macrophages and in immunocompromised patients can lead to granulomas. Some fungi produce toxins, commonly called mycotoxins. Aspergillus flavans produces aflatoxins. And you'll see this particularly when you're looking at the grains of peanuts, corn, other grains and nuts. But the aflatoxins can cause liver cancer. Let's finally go through some other eukaryotic parasites. It's very interesting because some of them have some very elaborate means to duck and bob and weave and still create infection. Most parasites either live in the intestinal tract or via and are transported via an arthropod bite, but the schistosoma species can bore directly into the skin. Most parasites attach to the host via specific receptors. Giardia limbilia has a huge disc on its cell to attach itself to the small intestinal lining. Hookworms use their jawed mouth attached to the intestinal wall, and that's basically how they feed on the blood from the capillaries in the wall. Plasmodium vivax, which is one type of malaria, attaches to the Duffy group of the red blood cell antigens. Parasites will evade the host immune system by entering inside the cells. So Plasmodium gets into red blood cells, Leishmania gets into macrophages. They coat themselves with host proteins, with schistosoma, and they change their surface antigens, trypanosoma for sleeping sickness. The parasite's damage can occur via the release of toxins when released into the blood, and this can trigger high fevers, such as with malaria. Usually what happens is that you're going to have the red blood cells burst, and they're going to release toxins and new copies of the malaria parasites. And that's going to lead to the fever and chills, etc., that we mentioned previously. Also, the parasites can enzymatically break down host cell tissues. Schistosoma is one, entamoeba, that amoeboid parasite, is another. Or they can block small intestines due to the presence of many tapeworms, or the granulomatous response, such as the accumulation of lymphocytes and macrophages in an attempt to form a wall around a persistent infectious organisms or antigens, such as to block off schistosoma eggs. Schistosoma eggs have a small little hook on them. They usually, what they do is eventually hook through and bore out of small blood vessels to get into either the bladder or into the rectum, and then are passed out of the body to set up the next life cycle of the parasite. Well, we've gone through a lot here, and what you want to do is take time to go over the microassessment, future opportunities, and the chapter summary. What I'd like to do now is jump over to the epidemiology, chapter 19. Oh, sorry about that. Now, a bunch of terms that you need to know. And this is really going to be helpful for your vocabulary when you're handling nursing, etc. Epidemiology is the study of the factors influencing the frequency and distribution of disease. Epidemiologists are health detectives that collect and compile data on the outbreak of the disease. A communicable disease is a disease that can be transmitted from one host to another. When we talk about the term reservoir, this is the source of disease causing organism. Example, rabies. Where would you think of rabies? Well, they could be raccoons, skunks, a rabid dog, but in Massachusetts, for example, every once in a while they have these massive upswings in rabid raccoons. And in the 60s and 70s and 80s, they had to develop strategies to try to stop that. In some cases, they would throw out bait via helicopter into raccoon-infested areas, and the bait would have rabies vaccine to it. This was just to damp down the rabies outbreaks. Portal of exit. This is the body surface or orifice where the pathogen is shed. Portal of entry is the place of entry of the microorganisms into the host. Now, a non-communicable disease is a disease that's not transmitted from one host to another. For example, histoplasmosis, H. capsalum, is transmitted via fungi held in dust from bat, chicken, or bird droppings. So in other words, if you go in and you're spelunking, or basically cave exploring, and you come in contact with a dusty pathway that basically is composed of a lot of decayed and ground down bat droppings, you breathe it in, you may get histoplasmosis. But you can't go home and then cough and then give it to mom, brother, sister, uncle, etc. Now, we have to start dealing with some of the terminology beyond this when we talk about the rate of disease in a population. When we talk about rate, we're talking the percentage of a given population infected, not the absolute number of cases. When we talk about the attack rate, this is the number of cases developing in a group of people exposed to the infectious agent. When we talk about the morbidity rate, this is the number of cases of illness in a given time period divided by the population at risk. Mortality rate is the death rate from the disease, so the percentage of population that dies from the disease. When we talk about the incidence, this reflects the number of new cases in a specific time period in a population at risk. Prevalence reflects the total existing cases, both old and new, in a given population at risk. Terminology endemic it refers to diseases that are constantly present in a given population. When we talk about the term sporadic, those are cases that only occur from time to time. Epidemic is an unusually large number of cases of an illness in a population. An outbreak is a cluster of cases occurring during a brief time interval and affecting a specific population. Now, the term pandemic is an epidemic that is spread worldwide. So what happens is, if you actually look at CDC, they actually have a listing, or WHO, and if there's an outbreak within a country, they'll give it a certain classification. If it spreads to several different, let's say, states or provinces, they will have another classification. If it jumps from one nation to the other, they will have another classification. And if it jumps across a series of nations on several different continents, then they would probably consider that pandemic. Okay, so it's how they use the terminology. By the way, what you're seeing here is epidemic thresholds, and you're seeing basically the incidence of pneumonia and influenza deaths. Okay, the percentage of the deaths over several years, and what they do at the bottom there, the x-axis, is they look at the weeks. And what you always see is a seasonality of occurrence. Okay, and the actual percentage of total deaths in the population. And so what you'll see is, when we talk about this, some diseases, some outbreaks are always connected to a particular time of year. Others are fairly consistent throughout a certain time of the year. Okay. Now, I've given you that handout on chain of infections. That is an extra handout for this week. Please print it up and review it. It's much more detailed than what you see in this minor image, but it could be very helpful in you reviewing in the concepts. Now, when we talk about any type of infection, it could be animals, humans, etc. It's very important, as reservoirs can resupply pathogens to hosts after long delays since the last outbreak of the illness. They can be controlled from having another outbreak due to altering the environment for reservoir organisms. Let me give you an example. Control rats, you control the fleas of the rats. You control the fleas of the rats, you control the pathogens in the fleas. But let me give you a couple of other perspectives here for a second. When we talk about reservoirs, right now in the world, they're concerned about plague. But there is a certain level of plagues in marmosets and in certain other mammals that are small, that are up in Central Asia. And so they periodically study these, just so they can understand how these small animals can retain and survive and have this particular type of bacteria. And so the bacteria may have set up itself with a type of host pathogen interaction that causes minimal damage to the host, but it can maintain and survive inside of the host too. When we talk about human reservoirs, though, this is a little bit different. Symptomatic infections, which depend on the cooperation of the infected individuals, the incubation time of the pathogen, the means to isolate individuals, and whether infected individuals remain as carriers, shedding the pathogen after the symptoms subside. That's one thing. Think about it. You're working at some place, somebody comes in, they've got a little bit of a cold, and sooner or later, other people will have, and perhaps that very same cold. But then we have to deal with asymptomatic carriers. They have no symptoms, but they have the pathogen active in their body. Individuals may shed the pathogens from feces, breath, saliva, semen, vaginal fluid, etc. Now I got another image for you. Now there is a reference on page 648 you could read, but this is Mary Mallon, who lived from 1869 until November of 1938. She was known as Typhoid Mary. She was in isolation at this point. Mary never suffered from the symptoms of typhoid, but Mary was an Irish immigrant. She came into the country, and at a certain point in time in lower New York, around the New York City area, they were having, in the boroughs, an outbreak of typhoid. They couldn't figure out who it was or where it was coming from. Now, Mary at the time was an immigrant with very little skills, and so she did what she did best, and she cooked. Unfortunately, as she cooked, she would end up contaminating the food with typhoid, and people were getting sick in various restaurants and in various country clubs and places like that wherever she worked. They finally tracked her down and they isolated her, and they concluded that she was it. Now, believe me, Mary was not happy, and she was very resistant, which is one of the other problems that can occur with when you find asymptomatic carriers. The story goes is that the public health wanted to basically put her away. They kept her for a year. They then tried to say, look at Mary, we're willing to allow you to go free, but you've got to not be a cook anymore. So they actually trained her to do sewing. And everything was hunky-dory. They hadn't heard from Mary in a while, let's say about a year or so. And then they had an unusual outbreak. And so public health went to the site, and they're trying to figure it out. And lo and behold, who do they see there? Hi, Mary. They knew it was her. She tried to escape. They arrested her. They went before the judge, and they said, Your Honor, we cannot have her doing such and such. So they institutionalized her for the rest of her natural life at a asylum so she could not contaminate and make other people sick. And the typhoid outbreaks stopped, at least in that particular situation. Non-human animal reservoirs. Well, these include situations where the animals with zoonoses, now that's a disease found to infect humans, but found primarily in animals. Usually zoonoses are more severe as humans have not developed the balanced pathogenicity with the pathogen, whereas some animals have that. And I gave you the example of prairie dogs with Yersinia pestis, carrying plague. And we also have to deal with environmental reservoirs. Some pathogens exist in the environment, such as soil, such as Clostridium botulinum, Clostridium tetani, which causes tetanus, Legionella pneumophilia, which is found, interestingly enough, in water coolant systems. So, now we can deal with portals of exit. Portals of entry, portals of exit. We've talked a little bit about entry, but what about exit? For organisms to leave the host and infect new hosts, they must have a means to leave the host. Many that inhabit the gastrointestinal tract are shed in feces. Others that are sexually transmitted diseases, such as Neisseria gonorrhoeae, can be carried on semen or vaginal secretions. We have the hantaviruses found in the droppings, urine, and saliva of deer mice. So a lot of these different means can occur. Some of you might remember in the 80s, it suddenly became a big problem for blood-borne pathogens. And when people would have an accident or they'd cut themselves and they'd be walking around in a public area like a shopping area or a mall or something, I remember one place there was droplets of blood throughout most of this store. And I said to one of the workers, I said, ma'am, you've got blood on the floor. She said, oh, okay, that's nice, and walked away. Now we would all be going, you're kidding me. But they didn't have full training yet in the control of blood-borne pathogens and how to decontaminate those sites. Now that blood might have been totally harmless, but no one knows. Now let's talk about transmission for a few minutes. A pathogen must be passed from the reservoir to the new host. Now, how does that occur? We can have horizontal transmission, vertical transmission. Horizontal transmission is the transmission of a disease-causing organism from one person to the other through contact, ingestion of food or water, or a living agent such as an insect. Okay? And you can see some of these cases straightforward. Okay? People sneeze. People will also use indirect contact through surfaces, doorknobs, telephones, okay? Handshake, oh my. Vertical transmission is the transmission of a pathogen from parent to child, such as from a mother to an infant during birth. Group B streptococcus can be passed through the birth canal, and pregnant women to fetus can pass on what is called congenital syphilis. When we talk about contact, we're talking about the transmission of a pathogen, usually involving some form of contact. Now, you have direct contact and indirect, and you see this pretty clear. Direct contact occurs when someone physically touches another. It can be a mere handshake, it can be something much more intimate, such as sexual intercourse. And due to the fragile nature of some of these organisms, they may die if left outside of the body, and thus must be transmitted via direct contact. One of the other means is fecal-oral transmission, transmitting organisms that colonize the intestine by ingesting fecally contaminated material. This can happen with E. coli, Shigella, things like that. Now, indirect contact, you have the transfer of pathogens via indirect objects. We get into the term fomites, which are inanimate objects that act as transmitters of pathogens, such as viruses and bacteria, including doorknobs, drinking glasses, keyboards, cell phones, clothing. You've heard a bunch of stories where some microbiologists have done surveys on just keyboards at work and the bacteria that they find there and the fungal spores and the viruses. Okay? We also forget that it is important to break the chains of infection by washing the hands. We now have made an industry out of the hand disinfectants and the hand wipe cloths. So we may actually also take the disinfectant sheets and wipe down the entire surface of a desk or chair or doorknobs. Okay? We also have to break that chain of infection to wash hands after touching a patient and prior to touching an inanimate object. Now you see the person in the center, the airborne transmission. This is where we get into droplet transmission. Transmission of an infectious agent through inhalation of respiratory droplets. One of the biggies, sorry about that, one of the big problems is when you sneeze. Droplet transmission is an important source of transmission. This is usually a lot more effective when you have densely populated situations, schools, barracks, crowded subways, okay? Crowded concert halls. It's important to cover the mouth during the sneeze or the cough. Then we deal with food and water. Animal products may harbor pathogens, okay? Handling contaminated products can transfer pathogens to non-contaminated foods. See this? Yummy. One problem. That's raw chicken. What does raw chicken do? Hey, it could have on it a variety of pathogens. If you take the raw chicken off the wooden table, continue to process your fruits and vegetables, then take the raw chicken, cook it, then put it back onto that wooden table, the pathogens that have been on the raw meat are now on the table, have now been transferred onto the vegetables and onto the cooked meat. And then the food-borne infections begin. Okay? Waterborne diseases can occur especially when the municipal water system distributes water to a large population and when there's a malfunction. It can also occur if the water supply is also the sewer for the local population. One of the big issues that occurs if you ever hear about a place having an earthquake and you see that the water is leaking up to the top of the surface, odds are those are pipes underground beneath the sidewalks, beneath the roadways. They have broken, and probably the same pipes that are broken have been the sewer pipes, which means that the drinkable water has now been risky and contaminated. Go get bottled water, folks. What about air? Respiratory diseases can be transmitted through the air. While large droplets drop to the ground surface, others may remain in the air and travel via wind or heating and air conditioning duct systems. You have to keep in mind something else. The droplet nuclei. Now, the droplet nuclei is in essence solid form. So when you sneeze and there's a particle there, what happens is eventually that particle that's partly liquid and partly has, a little bit of salts or whatever, and got a little bit of, mucus or whatever, it's got some viruses or anything, it will begin to eventually evaporate off the water and becomes a solid particle. Okay? This nuclei particle, can now contain several viable pathogens and be inhaled or picked up by the skin, the hand. You also keep in mind that other airborne particles, dust, dead skin cells, etc., can also carry pathogens. That's why if you go to surgery, you gown up completely, because you don't want to shed dead skin cells. They're going to contain bacteria. And you don't want to have that in an open wound where you're working on somebody. Anyways, let's talk about vectors for a few minutes. Yeah, vectors. Now, any living organism that carries a disease-causing microbe is considered a vector. And the most common ones are mosquitoes, ticks, and flies. There are two forms of vectors. Mechanical vector, which is an organism such as a fly that physically moves contaminated material, such as pathogens, bacteria, whatever, from one location to another. Biological vector are organisms that act as a host of a pathogen before it is transmitted to another organism. The pathogen can multiply to high numbers within the vector. To control some outbreaks, this requires the control or eradication of the vector. Now, I included some notes on this, and I tried to include some articles on this. It is really, really important if you take the time to review some of this. There was one paper that came out recently where they looked at the bacteria that they found on a fly. And you would not believe it, they found like 300 plus different bacteria on that fly that are shed when it lands. Some of them included Helicobacter pylori. So it's really, really nasty. Now, what is a biological vector? The organism acts as a host for the pathogen before it's transmitted to another organism. Now, the pathogen can multiply to high numbers within the vector. This could be viral. This could be bacterial. Okay? Portals of entry. When we talk about portals of entry, this is the places of entry of the microorganisms into the host. Portals of entry include the integumentary system, the skin, the respiratory system, the oral area, but also keep in mind some of these other ones, like I said, the eyes. Since colonization is a prerequisite for causing disease, the organism must enter the host at a site that favors colonization. The importance of the route of entry can be illustrated by Yersinia pestis, the plague disease. If the flea bites the person, then bubonic plague, causing the inflamed buboes, the lymph nodes, the mortality rate is 50 to 70%. But if the infection spreads to the lungs, you have pneumonic plague. And when that occurs, you've got nearly a 100% fatality rate. That is, in both cases, if it is not treated with like antibiotics or anything else. Other Enterococcus fecalis intestinal organisms, they're basically harmless in the intestine, but they can cause bladder infections if they enter the urinary tract. So moving from there, let's talk about the factors that influence the epidemiology of disease. Various factors play a role in the development of infection. Different pathogens have different array of factors. When we talk about virulence, again, we're talking about factors that enhance the success of the pathogen. And these include attachment to the host, avoidance of the host, immune system. When we talk about the disease triad, otherwise known as epidemiological triad, this is a formation in which you have pathogen, host, and environment. So on each side of that triangle, the pathogen, and then there is the host, and then there is environment. All of these factors come together and either favor or disfavor the successful infections and diseases that can occur. The other factors that influence the epidemiology of disease include the dose. the minimum number of pathogens required to initiate an infection. The probability of infection and disease is generally lower when an individual is exposed to a smaller number of the pathogen. There must be a minimum number of pathogen cells, or in other cases, pathogen viruses, to induce or produce enough damage to cause disease symptoms. We also have to keep in mind the incubation period, okay? When we talk about incubation period, this is the extent of the spread of the infectious agent is really going to be influenced by the incubation. For example, some incubation periods are very, very long and help to enhance the transmission of the pathogen. For example, HIV. If the incubation period is long, this increases the chances of an outbreak spreading beyond local borders. One way to think about it is also this way. Think about it. If you got exposed and then were sick the next day, depending on how sick you were, you might what? Stay at home. But if you were infected with something and you could spread it to others while you do not feel any symptoms, and that you have a long incubation period, then you are more likely to spread that disease, and it is going to spread to many other individuals. Another factor is the host or population characteristics. These are certain population more likely to be affected by a given pathogen. The characteristics that influence the occurrence of the disease include immunity to the pathogen, general health, age, gender, religious or cultural practices, genetic background, and then we have herd immunity. This is the phenomenon that occurs when a critical concentration of the immune hosts prevent the spread of the infectious agent. That's why vaccination has been so critical, because the more of the population you have immune, the less chance you're going to have serious outbreak. Now, of course, there are some environmental characteristics too, and these include these influence epidemiology. They include the physical environment, temperature, water, which, for example, humidity, nutrient supply, and radiation. The human factors include crowded conditions such as, poverty, etc., poor sanitation. Usually, when you had certain disasters and the sanitation has gone from very fine or very good to really poor, that's when you can expect outbreaks, okay? Even in the United States, when they've had, let's say, a massive disaster like a major hurricane, and it's knocked down the sanitation, they know pretty quickly afterwards they're going to face some serious diseases that may be waterborne or may be related to the failure of the sanitation, removal of trash and garbage and things like that. Okay? So I want you to keep these issues in mind. Think about it the next time you see some news items about a disaster, whether it's in Latin America or the Caribbean or the United States or Asia or Africa, etc. And watch how the news changes from the immediate casualties to the picking up of things to whether they can keep from having outbreaks. Wars are another area that's big, in which a lot of times what kind of follows after a place has been war-torn is outbreaks of different diseases, in part because of crowded conditions and because of poor sanitation. And that includes even, yes, the poor capability to dispose of bodies because of the wartime situation. Now let's talk about epidemiological studies. And what we do there is there are epidemiologists that investigate a disease outbreak to determine the causative agent as well as its reservoirs and route of transmission. They may also recommend methods to minimize the spread of the disease, and their various studies are used to obtain different pieces of data. To help you think about it, keep in mind they're not just sitting there and saying, okay, now we know how this cholera outbreak occurred, but what they want to do is be able to provide some answers to say, this is how we've got to have it stopped. What you see here is a comparison here where this is an outbreak of tetanus, all right? Look at the number of cases. Look at the age groups. This is a histogram. Now, this was from a previous edition, but it's so perfect for what I want to talk to you about for a second. Most little kids get tetanus vaccine. most other people get their tetanus vaccine. But then when we start getting to be adults and when you start bopping around all over the universe, we think we're immune. Wrong. Tetanus is good for about 8 to 10 years. Then you got to get a booster. So why is the 25 to 39 and 40 to 64 group some of the highest number of cases of tetanus? Because they've not gotten a booster. They've not given themselves the booster. They've not maintained their immunity. So this is an epidemiological situation. And you notice that one of the factors is age. Here you have comparisons of propagated versus common source epidemics. The propagated epidemic there on the top, and then over a common source epidemic where you have a central exposure, and then you have an outbreak and then a peaking down, okay? Now, descriptive studies. These are the type of studies that seek to characterize a disease outbreak, and they determine the characteristics of persons involved and the place and time of the outbreak. Each factor provides important information. You have to ask a lot of questions when you go out into field epidemiological studies. The person, what was their occupation, age, sex, habits, etc. The place, this provides information on the site, as it may also include information about disease reservoirs, vectors, boundaries of transmission. Well, here's a question. Why isn't malaria occurring in Alaska? There's a lot of mosquitoes, but the mosquitoes cannot carry the malaria parasite. Also, the outbreak of mosquitoes is only a short 6-week period, and everybody else is covered very heavily before and after that short summer season. Time. When did the outbreak start? What season of the year? And a lot of these other factors play a role. You notice here, respiratory syncytial virus and Campylobacter. You notice some relationship here? If you note that respiratory syncytial virus seems to occur during the winter months and dies out during the summer. Campylobacter is the opposite, occurs more during the summer than any other time of the year. Reason? Well, Campylobacter is very common for food-borne illness. And a lot of people go out and do picnics and backyard barbecues and things like that. People are contained inside, and if they sneeze or pass on the respiratory syncytial virus, mostly when they're enclosed is the place and time and conditions that favor RSV. Okay? Common source epidemics, outbreak of the disease due to contaminated water or food or single source of an infectious agent. If you ever get time, review some of the stories of one of the first real epidemiologists, John Snow, and how he was able to understand the outbreak of cholera in, I believe it was early 1800s, I think it was, of London. And all he had to do was have the handle of a particular water pump removed. The handle was the formite that allowed for the passage of disease for all the people getting the water there and passing on the disease that killed so many people with cholera. Okay? Propagated epidemic. This is an outbreak of a disease in which the infectious agent is transmitted to others, resulting in steadily increasing numbers of the people getting ill. And if you go back to here, you see this. Where it's propagated, it starts having an incubation period, and so you get these waves, and it keeps going up and down, depending on the number of people and number of cases. Okay? So, let's move from here to analytical studies. Now, when we get into analytical studies, oh, and by the way, I included this other one here to show you seasonality of some outbreaks. E. coli O157, Salmonella, Campylobacter, all the same thing. Outbreaks are highest during the summer. This brings us to figure 1911, okay? When we do analytical studies, this is a type of study done to identify specific risk factors associated with the development of a certain disease. Now we have to understand what risk factors are. These are specific conditions associated with a high frequency of the disease. When we do cross-sectional studies, this is a study that surveys a range of people to determine the prevalence of the characteristics, including disease, risk factors associated with the disease, and the previous exposure to the disease-causing agent. Then we get into these other studies called retrospective studies. This is a type of study following disease outbreak that compares the actions and events surrounding the clinical cases with those of controls. So in other words, if you go back to the very beginning of the chapter, what did you find out about? You found out a little bit about, if I'm not mistaken, yes, the glimpse of history was Semmelweis. And Semmelweis was the one that helped to conquer purtibuvule fever. And this was caused because doctors did not wash their hands, literally. They would do autopsies and cut stuff and everything else and come in and try to deliver a baby. And the women would get sep infections afterwards. It's very fascinating, and there are books out about his life. He really helped to save so many lives, and nobody really gave him credit for it for quite some time. So those are the retrospective studies. They compare the actions of what they did with those that were the controls. Okay? Prospective studies look ahead to see if the risk factors identified by the retrospective study predict a tendency to develop the disease. So these would be sitting there going, what happens later on? We also have to deal with what we call cohort groups. These are populations with a known exposure to a specific risk factor that is followed over time in a prospective study. A cohort group could be all teenagers, all athletes, all football players, all police and law enforcement, all high school students, etc. Okay? And you have a very nice case presentation in 19.1 that will give you some type of example, I believe. And then we have experimental studies. This is a type of study done to assess the effectiveness of measures to prevent or treat the disease. Now, when you do these ones, you have to include the placebo, which is sort of the control, but it's really a sham treatment. So if I'm going to give people a drug, and then I'm going to give the others what looks like a drug, but it's really a placebo, a sugar pill. Placebo is Greek from I come to please. And so what you do is with these experimental studies is you have individuals that are treated with and those that are treated without, and find out what's the end result of the treatment of the disease. Now, a double-blind study is an experimental study where neither the physician nor the patient knows who will receive the real treatment. Usually what happens is you record the data, you give the people what looks like a pill. It could be the sugar pill, the placebo, or it could be the actual drug. And at the end of the study, someone comes in and breaks the code and determines which ones were the treated group, which ones were not, and then you publish the work. Okay? Now, infectious disease surveillance. This is where we get into table 19.1. You didn't think that this occurred, did you? Well, it does, and it's a lot. Now, oops, I went backward here. Sorry about that. Let me help you here. There is an infectious disease surveillance network, and this is critical, both nationally and globally. When you talk about national, that's the Centers for Disease Control and Prevention, CDC. This is absolutely essential in our global society that we have these infectious disease surveillance networks now. This provides us warning if there's going to be serious possible outbreaks or epidemics, and they help us with disease prevention. Now, CDC is part of the United States Department of Health and Human Services, okay? So DHHS. They collect data on diseases of public health importance. They publish reports of outbreaks and disease research. And you can see the table 19.1. These are all the diseases that they have to report on. Now, just to help you, you can get emailed to you copies of the MMWR. I know some people might say, no, I don't need any more boring email. But the reason that you have to understand that this is available, and it is weekly. Congress mandated CDC to report occurrences of over 50 diseases. And most of this data is provided by the state public health department. So the Mass Public Health Department provides for what goes on in Massachusetts, passes it to CDC, they compile it and release a report every week. Okay? And this is really interesting because this is where you can see, hey, is there going to be an outbreak of this, or is this quite prevalent, or is this something new that's showing up on the radar screen? There are public health departments, the state agencies that include public health laboratories, okay? Some states have mandated that physicians report certain communicable diseases to the state public health department. Some of these state labs also provide testing for physicians, for public water supplies, etc. Some of these labs are also the same labs that if they come in and say, they're brought to them, we have this material, we think it could have been a bioweapon, could have been an infectious agent, they will do the testing there, and then they spread the information and pass it back to CDC. Okay? Now, the big also international agency is the World Health Organization. This is an agency of the United Nations, and it serves 4 functions: provides worldwide guidance in the field of health, sets global standards for health, strengthens national health programs, and it develops and transfers appropriate health technology. And they also disseminate information through publications. You can see them on the internet. You can have them delivered to you by email. One of the things that happens is that WHO and CDC work very tightly together when we have things like bird flu possibility, novel influenza outbreak. You see a lot of activity. And if they see that we're getting to the possibility of an epidemic, they start setting up all the mechanisms and warning, etc., for the possibility of how to deal with it, and yet not, and also to try to minimize the pain and suffering and deaths that might come from this outbreak. Now, there are some trends in disease that you need to be aware of. See this poor little boy here? This is a case of smallpox. Now, smallpox doesn't exist anymore, Dr. Roberge. It's a disease that's been pretty much eradicated because the only reservoir is human beings. And it's pretty sad. If you look at a variety of books, one of them called Scourge, Jonathan Pollard wrote a couple of books, and there are also Henderson wrote a book on his efforts to have it helped eradicate it. When health surveillance and departments of health, nations all work together, you would be surprised what can actually occur. This disease that's been around since human beings were around was eradicated. There are only 2 places that are known, confirmed to have repositories of the disease left: CDC and Vector. These are research labs that have it, and they've gone through the genome, they've mapped it out. But sadly, there are fears that somehow out there, some people have decided to make this a terrible bioweapon. And as we all get older, and I know I've got my smallpox vaccination mark, most of the population is not vaccinated against it. There are some that are vaccinated because they go into the military. But this can be a very debilitating disease if you get sick with it. And I'm sorry about my dog in the background. Please bear with me. But the thing that you have to understand is this. How this disease was eradicated was combined efforts of a lot of physicians, epidemiologists, doctors, people on the ground, and the work was done under the World Health Organization. And by 1977, they had eradicated it. Now, polio, measles, draculiasis are targeted for eradication as well. Draculiasis is known as the guinea worm, or basically it's a terrible parasitic disease that goes underneath the skin. Measles, many people know it's a viral disease, and so is polio. They're all trying to eradicate those. Now, emerging diseases. We have great challenges in trying to reduce emerging diseases. This is by definition infectious disease that is increasing incidence and geographic range. And it's due to the microbial evolution and mutation. It's also due to changes in human behavior. Populations moving into areas that were previously wooded, forest, jungle. They were never occupied by human beings, so humans never had any contact with some of these pathogens before. But emerging diseases can also be as a result of war, civil unrest, as I said, expansion of populations in new environments and global markets. New diseases and new outbreaks are going to continue. That's just the way it's going to be. In a globally active world, the global marketplace, we are going to stretch out and interact with lots of new ideas and resources, and in some cases, we're going to meet up with some new health challenges. That's why this is also an interesting field. If you like a challenge, this is the area to go. The contributing factors to the emergence of these diseases include microbial evolution, complacency and public health effects, changes in human society, the rise in daycare, for example, and the rise in technology, population expansion, urban development, mass production, widespread distribution and importation of food. I mentioned to you before how you had some cases of a protozoan parasite that showed up in a wedding party in a fruit bowl dish, a big one for a party of 200, and they tracked it back to raspberries raised in Guatemala, and somebody there had a bowel movement in the fields and as a result contaminated the food, and nobody knew about it till far, far later. What will also occur are war and civil unrest. Just can't help it. You start bombing out situations and places and infrastructure, you tend to start creating extreme poverty, destruction of basic water transportation, food transportation. The cholera outbreak in Yemen right now is caused because of the Yemeni civil war and the war that's also going on between the Houthis and the Saudis. And this is at the beginning of 2018, but this is something you need to know. As well as climate changes. Okay, climate changes. Now, as we talk further, healthcare-associated infections, and by the way, healthcare-associated infections, HAIs, used to be known by another name, nosocomial infections. What these are basically infections acquired during hospitalization, usually due to patients recovering from another illness or injury. The infection depends on the length of time exposure, virulence, and the number of organisms, and the state of the patient's host defenses. By the way, it doesn't just have to occur in hospitals. It can occur in therapy centers, urgent care centers, long-term care centers. It can range from mild to fatal. The organisms can occur in the normal flora to imported organisms, imported from other patients or present in the building environment. In some cases, hospitals have had to tell visitors, you cannot visit anymore our patients in here. And the reason is they have so much of influenza going on. Or I knew one case, a young man was recovering from an injury in his leg, and the kid next to him in the next bed had a very larger family, and the larger family came and visited. And when the first kid got home, after a certain period of convalescence, got a letter from the hospital saying, you need to have your son checked for TB. It turned out that some of the relatives that visited the boy in the next bed had active tuberculosis. So you can see that this gets to be a tricky situation. Okay, so we're moving by here, and we see the relative frequency of different types of nosocomial infections. Some of these pathogens, I would refer you to table 19.2. Now, you don't have to memorize the whole list again. This is not something where I want you to memorize it all, but get somewhat familiarized because we are going to come back to these in various situations down the road. Enterococcus species, that's usually from intestinal flora. Escherichia coli, these are the Enterobacteriaceae. And Pseudomonas species, these are from moist environments. Staphylococcus aureus, you can find these from formites. And Staphylococcus other than S. aureus You might find these in the biofilm forming from things like indwelling catheters, etc. What are some of the other reservoirs? Well, that can be other patients, the hospital environment, healthcare workers, and the patient's own normal microbiota. Transmission in hospitals can happen from a lot of different means, medical devices, healthcare personnel, or it can be airborne. That's why it's almost like counterintuitive. Why? In a healthcare facility, people are getting infections. Well, that's because sometimes it's brought in. That's because the patients are weakened. That's because of a variety of other problems or challenges that occur. And so we need to be aware of these, and we need to be able to prevent them. Usually, prevention is overseen by an individual specially trained in this, called the infection control person or the infection control committee. They usually have a couple of people. You may have a hospital epidemiologist who's specially trained in hospital infection control. You may have an infection control practitioner, a specialist who performs active surveillance of the types and numbers of infections that arise in the hospital. Now, as we're starting to wrap up, I did want to bring your attention to perspective 19.1. Okay? Maintaining vigilance against bioterrorism. Part of this course includes the reality that we are going to have this. And therefore, here's what I encourage you to do. Review the future opportunities. Know the definition of bioterrorism, deliberate release of infectious agents or their toxins as a means to cause harm. Now, I don't expect you to memorize all the agents of bioterrorism, because guess what? You'll bump into a bunch of them as you deal with some of the diseases. But some of them I want you to be aware of. And those include Francisella tularensis, that causes tularemia, Bacillus anthracis, that causes anthrax, Yersinia pestis, that causes plague, botulism, which is basically Clostridium botulinum. Okay, that's the toxin that comes, and of course, smallpox. Now, you might sit there and argue with me saying, I am not going to be a bioweapons specialist, etc. You're going to be a healthcare provider, and sad to say, this may come where you're going to have to face this. As the patients are streaming in, the surge comes in, okay? Also, I would encourage you to do this. Get familiar with just the CDC bioterrorism categories, A, B, C. They're relatively straightforward. Basically, which ones have the highest risk, and it's partly because they're easily transmitted, okay, and they can result in high mortality. B is moderate risk because they may be easy to spread and cause moderate mortality, okay? And if you look at a bunch of them, they're not that difficult. And by the way, it doesn't have to be nation versus nation. If you look at what happened of the Bagwanish cult in, I believe, the Dalles, Oregon, in the 1900s, I believe it was in the 1980s, they were a cult and they wanted to force the rest of the community to adapt to their restaurant. And so they basically made, are you ready? Typhoid fever bacteria and put it into the salads stations of the competing restaurants in the town, and a bunch of people got sick. Okay, and so this was a small group that did this. They developed the cultures and spread them out, made people sick. Also, category C, which is basically emerging pathogens. Some could be engineered for easy dissemination, and some of them would have a major impact on health. Aside of that, I encourage you to review the chapter summary, and I hope you all have a very nice week. We're just going to pass over that, and have a nice week. We will see you next week.